Hi, my name is Jason Graff. I'm a phytoplankton ecologist and oceanographer at Oregon State University in the Department of Botany and Plant Pathology. I'm currently part of a large project funded by NASA called Exports that's looking at the transfer of organic carbon that's made by phytoplankton in the surface ocean and how that is moved into the deeper interior ocean and used by the organisms there. Some of this carbon ultimately ends up in the deep, deep ocean where it can be stored for many thousands of years. As a plankton ecologist, I'm always amazed at the size, shapes, and diversity of phytoplankton and other plankton that we see in the water column. These single-celled plants are microscopic, some as small as a bacterium. These single-celled plants are extremely productive and produce the vast majority of food and energy for the organisms living in the ocean. Because of their abundance and how they interact with light, we're actually able to see these cells using optical instruments, including satellites. In these images, we can see the distinct colors of the pigments inside of the cells. These cells contain chlorophyll A, much like the plants on land. Chlorophyll is a distinguishing feature of phytoplankton in the ocean and is one of the properties that can be detected by satellites. This gives us the ability to make maps like the one shown here that provide us deep insight into the distribution and concentrations of phytoplankton and the carbon that they carry. In this time series of satellite images collected over multiple annual cycles, we can see how these phytoplankton change in time and space. In this time series, the dark blues and purples represent low concentrations of chlorophyll, and the greens and reds represent very dense concentrations of chlorophyll or phytoplankton. My role in the NASA Exports program is to look at phytoplankton, look at their abundance, their sizes and shapes, and try to draw relationships between the optical properties we see in the water as well as above the water. To do this, I use state-of-the-art technology. Here I'm standing in front of an instrument called a flow cytometer. It's a fancy instrument that lets me look at individual cells and their optical properties. We also lower optical instruments into the water to look at the bulk optical properties and try to relate those to what we're seeing in the flow cytometer and other measurements of phytoplankton that we can make. We also conduct experiments where we manipulate the environmental properties in which the cells live to see how they respond. These changes in the properties of the cells can then be linked to environmental parameters and furthermore the optical parameters that we can measure from satellites. These results can then be shared with our collaborators from other institutions who are working on critical aspects of the carbon cycle relative to the NASA Exports program. You might be wondering how I, or anyone for that matter, gets involved in oceanography and finds that as their career. When I was younger, I didn't realize that oceanography was a potential career. It took me a long time to actually find it. I moved to Seattle, Washington after I got a degree in advertising and worked in that black building in the center of the picture here that looks like Darth Vader built it. After a while, that field wasn't very satisfying for me and I knew I had to change my career path. After doing some research, I found that the University of Washington in Seattle offered a degree in oceanography. I registered, completed my bachelor's, and later got a PhD. And now when I leave the office, instead of wearing a shirt and tie, sometimes I look like this. And I'm fortunate that when I'm not at the office, I get to go out to sea to do my work, have meaningful conversations with colleagues and friends while looking at the ocean and hopefully understanding it better.